Today we will be talking about DNA sequencing, a method used in the Human Genome Project, and we will relate this to a lab we did in biology class known as the Bacterial Transformation Lab. I'm Suhas Nandiala.
And I'm PGLo.
The Human Genome Project started in 1990 and ended in 2001. Scientists originally predicted that it would end in around 2014, but it finished 13 years ahead of schedule. The government completely funded this project, and it had two main objectives. The first objective was to map a complete human genome, or figure out all the genes that you could find inside a human body. The second objective was to identify the functions of all these genes that it discovered.
So the outcomes of the project were multifarious. We mapped out about 20,000 genes of a person. We also determined that 99.9% of all nucleotide sequences are the same for all humans. We also discovered the functions for 50% of the discovered genes. So for the human genome project to work, we needed to find an efficient way to sequence DNA. To do this, we use something called the Sanger method. First, we insert dideoxynucleotides that terminate replication of DNA.
How this works is that let's say we have DNA replication going on. In this case, let's say we have an adenine that needs to bond with the thymine. The thymine would be missing an oxygen because it is a dideoxynucleotide, so it would not be able to bond with the adenine because it does not have an oxygen and hydrogen. So bonding is not possible. So it will terminate the replication of DNA.
Also, we used a gel that had 4 lanes. We put DNA base into each of these lanes, and we inserted a negative electric charge. Since DNA is also negatively charged, these two things will repel, and DNA will move down the gel. We manually read the position of each of the bases, and that would be the DNA sequence. However, manually doing it was very inefficient. So we started putting dye labeled bases instead of normal bases. Then a computer could read the dyes and identify the bases much more quickly and efficiently.
In class, we did something called the Bacterial Transformation Lab. This lab was a genetic transformation. Our task in this lab was to insert a gene that coded for green fluorescent protein, or GFP, that caused the bacteria to glow. This gene that coded for GFP originally came from jellyfish. Now, with the help of Luria broth, our task in this lab was to insert a gene that coded for green fluorescent protein, or GFP, that makes the bacteria glow. This gene originally comes from jellyfish. With the help of Luria broth, or food for the bacteria, a DNA plasmid, ampicillin, an antibiotic, and arabinose sugar, the genetically transformed bacteria was able to glow. On the left-hand side of your screen, you can see 4 agar plates. 2 of these agar plates are marked with minus DNA, and 2 of them are marked with plus DNA. The plus DNA contain plasmids, and the plate on the very right side contains Luria broth, ampicillin, and arabinose. This plate resulted in bacteria that glowed, as you can see on the picture on the right. The plus DNA with Luria broth and ampicillin without arabinose just created many colonies, but didn't glow at all. The plate with minus DNA and Luria broth just created one large clump of DNA, and it didn't make any colonies. The minus DNA with Luria broth and ampicillin killed all the bacteria, and none of it existed on the plate anymore.
So there were many relationships between, like, we had to map the genes of a human in the Human Genome Project. For us to identify the gene that codes for GFP, we also had to map the genome of a jellyfish. However, we didn't have to sequence all the genome of the jellyfish. We could use reverse engineering of a protein, of a GFP protein that already existed in live jellyfish, to reconstruct the amino acids and replicate the protein. Thanks for watching.